As I noted in the previous video, the process of replicating DNA is quite a complicated one, and that's true chemically and biologically as well. I mentioned also that I don't want to get into too much detail about the biology because this is a chemistry class, but I would like to make sure you understand the function of some of the protein factors that are involved with this entire process. Let's start at the bottom of the page where we were working on Okazaki fragments, and let's again have a bubble that shows anti-parallel directionality, that has RNA primers in it, and we're ready to replicate. First of all, I want to tell you that the primers are added by an enzyme called primase. This is what we call an RNA polymerase, and it comes in and uses nucleotides to synthesize about 4 or 5 bases of RNA. As we pointed out in a previous video, the RNA primers are going to be extended with DNA, and they're going to do so at the 3' end. So the arrowheads I just added are 3' ends that are going to extend. Now, the DNA itself is going to be added by an enzyme called DNA polymerase. So let's write that name down. It's an enzyme that makes a polymer, so we call it DNA polymerase. Sometimes it's just referred to as DNA pol. In fact, different textbooks say different things, but many say that it's the 3rd version of DNA polymerase that does the synthesis of the leading strands. And those are going to be identified as DNA pol 3. Now remember, we have Okazaki fragments or lagging strands that are synthesized as well, and those are made by a DNA polymerase as well. They're made by an enzyme called DNA pol 1. And again, different textbooks will tell you different things. So we're just going to agree on these things and understand that it's complex and very difficult to sort out. So this is going to get us a good chunk of the synthesis made within the bubble. Now, remember, as we continue on in this direction, leading strands can keep on polymerizing by DNA pol 3. Forgot that one down there. And in order to do so, they have to unwind the DNA. We've got it shown as double-stranded DNA outside the bubble, but in fact it's actually twisted up. And we need a couple of factors for that. One of them, I'm going to draw like a wedge, as if this wedge is forging ahead in both directions to unwrap the DNA that's all wound up on itself. These are called, there's actually enzymes, they're called helicase, and they need ATP as an energy source to unwind the DNA as the polymerase action proceeds toward the end of the chromosomes. Lastly, because the DNA is all wound up on itself, we need to consider that it gets over-twisted and we have to relieve some of the tension. And so there is an enzyme that cuts the DNA. We call this gyrase. At least in bacteria, that's what it's called. Sometimes called topoisomerase. Again, there's different variations and different books say different things. But these enzymes break the backbone and relieve tension that would occur as you try to unwind the DNA. This is related to supercoiling of DNA. So, lastly, we have to talk about a few items that are a little bit new, and that is to do with nucleases. So, nucleases break off nucleotides from DNA. Let's give ourselves a little more room here. That is, they break phosphodiester bonds, PDE, bonds that make up the backbone. And why would we want to do that? These polymerases are going along and synthesizing DNA. Why would we want to actually break it down at the same time? Well, here's the interesting take on this. Polymerases do make phosphodiester bonds, but the different polymerases do different things. DNA Pol 1 has what's called a 5' exonuclease and a 3' exonuclease. Let's take this apart a little bit. 5' and 3' are the termini of nucleic acids, and exonuclease means chewing up DNA or RNA from the outside. Exo means outside, like an exoskeleton. And what we're going to be seeing is the activity. Let's take a look at DNA Pol 1 here. Let's say it's cruising along here making DNA. Well, remember, the RNA has a 5' terminus right there that the DNA polymerase is ultimately going to bump into. So the 5' exonuclease is going to chew the RNA up from the 5' end, and we need to note that. This removes RNA. Now, remember also that the DNA that's being extended is a 3' end. That arrowhead right there of the blue fragment that's being extended is 3'. Why would we want to chew backwards and chew from the 3' end and take away what we just synthesized? The answer for that is for editing, and we call this sometimes a proofreading exonuclease. So what we have here is kind of like a backspace key. Think of how it is when you're typing and you're typing along in making words. If you make a mistake when you type, then you use your backspace key to back up, and that's what's going to happen here. DNA polymerase is going to back up and chew up anything that it did incorrect, and then it's going to proceed forward again. And in the meantime, it's going to use its other 5' exonuclease to chew up that. Okay, it's also worth noting that DNA polymerase 3 has to do proofreading as well. So it has a 3' exonuclease activity for proofreading, same thing. Just imagine what we're saying there. I'll just show you again. As the DNA polymerase 3 is kicking along there, it needs to synthesize DNA in this direction, and it needs to back up and proofread in this direction. However, DNA Pol 3 never is going to run into the primers because there's no primers ahead of it over here. So therefore, what we can say is that DNA polymerase 3 does not have the 5' exonuclease that's needed for removing RNA. Okay, one last factor. I've run out of colors. We'll go on to a different color. DNA ligase. You may have heard this in biology, but let's go back up here and imagine DNA polymerase is cooking along, making DNA. It's also chewing up the RNA. When it gets back to DNA, after it's removed this red RNA here, suddenly it bumps into the DNA, and at that point we have all DNA, but it's not sealed together, and DNA polymerase can't do that. So the DNA ligase is going to seal the nicks between the fragments of DNA. There's a lot of fragments here that are a lot of factors here that are involved, and we just really want to focus on their function and how they fit in to the action that's occurring inside this bubble. That's the end of this video.